Hey, so it's first of all, it's nice to meet you. It's an honor to meet you actually. I was doing some reading on, your background and the research that you did, and
Mhm,
I was one of the things that shocked me was how much resistance that I guess pushback there was towards,
mhm,
your discovery, which to me is one of the, I mean, now that we're looking in retrospect, it was a very progressive way of looking at vaccines. And
mhm,
two, I mean, I've run trials that incorporate your technology, as far as like mRNA vaccines. So
mhm.
I was, as far as one of the questions that I had for you is, where do you think the future of mRNA vaccine technology is headed? And especially with, like AI integration and if that's something that's even being looked into for the identification of certain nucleotide patterns and cells.
So, might mention about the resistance. I have to say, are you a molecular biologist yourself?
So I'm more on the neuroscience side, but
Yeah.
currently I
Mhm.
do some clinical trials with
Yeah. Mhm.
more so on the GI side.
Yeah.
But
Okay.
yeah,
But if you ever work in a molecular lab, we work
yes.
plasmids. And to, plasmid, it start everything with this DNA, which you grow in a bacteria. And then you have to isolate back, yeah, to purify the plasmid. And you start with adding RNAs if you use the quietine kit or something. And the laboratory, all of the molecular laboratory is contaminated with RNAs. And when I told somebody that, I work with RNA, and those people had experienced difficulties to isolate even RNA and hated the RNA work
Yeah.
because it always degrades. And they thought, oh my God, I hate RNA. Well, how you can work with it? It always degrades. So that's what they assume, that it is so labeled, but because everything is contaminated already on those laboratory, the jail equipment, the refrigerator door, and everything, and whatever they touched, it was RNA disappeared, and so that's what they were skeptic. And
Yes.
I could see the data, and so I was keep pushing forward. And so, of course, you can see that more than 150 clinical trial ongoing right now using messenger RNA. Half of them is vaccines, mostly viral. So prophylactic vaccines for things which has not existed, like HIV and influenza vaccine is developed. So some of them is we had, and
Mm-hmm.
shingles they try to develop, and others.
RSV.
So that RSV is already approved, and others is bacteria, so against bacteria. So there are like tuberculosis, or Sanofi has like acne for, has a clinical trial ongoing, and parasites like malaria sponsored by Gates Foundation. So that there are different pathogens, they try to prophylactic vaccines or therapeutic vaccines. The others is against cancer is mostly, using mRNA vaccine as a cancer vaccine. Those are individualized cancer vaccines, so the patient's tumor is sequenced and finding a mutation, and then those mutation incorporated to the mRNA that would code for those certain proteins with the mutation. So that's how it is developed. And they give back the patient like weekly dose. And so they can prevent return. And that was, BioNTech was the one for the pancreatic cancer, run this trial, and more than 3 years not returned. The cancer, Moderna run for melanoma, for example, some individualized cancer. There are also shared tumor antigens where, whatever tumor type they can give to the patient. And I also was helping to develop mRNA coding for cytokines, which can stimulate the immune system, or mRNA coding for antibodies so that the body can make the antibody, which is otherwise expensive medicine. And you can see that they are using for when somebody has a disease and not enough protein is made, so they deliver the mRNA because it continuously, maybe they can have for a protein for a whole week instead of daily injection. And the latest one is really the autoimmune disease. They generate CAR T cells, and for myasthenia gravis is already phase 3 mRNA-based CAR T cell therapy, and also in lupus they have clinical trials. So many of them is running actually in China, because right now in here is not favored the messenger RNA, but science will win no matter what.
We know that for sure. Science will win. I agree with you there.
Always. And a lot of the trials that I've worked on I've seen a shift from focusing on attacking the symptom to a more prophylactic approach, as you've mentioned, to where they're kind of trying to go further up the cascade to the nucleotide sequence, to the RNA, as opposed to, like, even with MASH drugs or Hep C drugs, you're starting to see trials that are looking at working on the modification of the nucleotide sequence, as opposed to trying to get rid of scarring or things like that. So it's very interesting to see how your thought process is continuing to permeate this space further than maybe even intended, but into areas where people weren't even, beyond vaccines in general. So
Yeah,
that's amazing
I
work.
worked also actually TGF beta 3, which can, as a protein, was already introduced, which makes the scar free, healing. And I generated mRNA for that. And others are doing, many things. I can see, okay, I tried that and I submitted grants. I never get any of that. But I can see now that companies are formed for that, and they are doing, and I am very happy. And better understanding the disease, because many times you are just treating the symptoms, but you don't understand really that what causing it. So that's a lot of things for the new generation of scientists is waiting for, that they should discover that what causing certain diseases.
Yeah. Katalin, I'm sorry, I have a question that's come up while you were talking. Those are such good questions, Ramesh. I'm curious, is there a team messenger RNA versus team altering DNA permanently, like the CRISPR? Is there a weighted push for where we are today and moving forward for either or?
Yeah, I mean, they are using actually the messenger RNA coding for Cas9 enzyme, and Intellia has a clinical trial running for transthyretin amyloidosis treatment, and those are advanced, I don't know, phase 2 or phase 3 trials, so that they permanently can change the genome. So interestingly, because everybody, when there was gene therapy meeting, I remember in the 90s or 2000, I went, nobody was talking about messenger RNA, maybe DNA, but even plasmid was not really. And because, the gene therapy has to be the DNA. And then turn out that even for gene therapy, maybe the solution is mRNA, because, the Cas9 is transiently present. So they are using for it.
Katalin, I bought Ramesh because I want him to do PhD. So you're talking with you, inspiration for you to do PhD program. That's yeah. Thank you for Ramesh. You have any questions, Ramesh? You're done, or you have
So the last question I have for you is, for what, and this is I guess more philosophical / existential.
Okay.
What pushed you forward intrinsically when you were getting pushback for trying to push your research, when it might have just been at a certain point just you believing in it, or but like, what was the intrinsic thing that was like, I know this is correct, I have to keep fighting for this?
The data. I could see that, and I could see constantly how improved, less RNA, more protein was produced, and the data So it was, I believe that, what I was doing is, and because I did with my own hands. I had no technician ever. I made all of the solution I made. I was sure that zillions of mistake I have not made because I paid so much attention, and so I was believing that what I am doing, and I did not. So I had to also convince one person, at least, who had money, and that was first Elliot Barnathan. 7 years he paid my salary. I was first on the faculty. I supposed to get my own, but I didn't get my own money. And then later, David Langer, who was a resident in neurosurgery, he convinced the chairman to give me space and salary. So I was employed by neurosurgery department for 17 years. And every time, they try to eliminate me, but, then David appeared from somewhere and then made sure that I will not be kicked out. And so I had to have somebody who had the funding because I did not. And then is this conviction, that the believing and I was not. So somebody was criticizing. I was always listening. But I knew that they don't see the data and they were not willing to listen to me. Then, I did not take seriously the criticism. You have to also understand my superior, actually, in cardiology was, very mean. And so if you respect somebody and then says something very detrimental, you are stupid or something, you have to take that person out of the pedestal. That I cannot respect, because otherwise you are destroyed. So for me, it was always important, this mental health, that person is not important for me anymore, even if it is my superior, because that how you have to learn handle the stress. And that's what I learned at age 16 by studying a book with our teacher in high school, that how to handle stress. And that if people would look at the same thing, same difficulties, but look at differently and act differently, they would be much happier life. And we practice there, even the very simple things, Ramesh, I would tell you that, hey, you are stupid. And then later I regret. And what the people are doing, they agonizing on it. I shouldn't say, but yeah, Ramesh said something which was really stupid and he started, and they wasting their time. What we learned is that I have to go, Ramesh, please forgive me. And that's it. If you forgive me or not, I cannot depend on your decision how I feel. So we learned that, okay, we have to act. And when I go back to you and say, I don't start with, hey, forgive me, but really you started because no. So and it is so simple. And we practice in high school that's how we have to act. And we don't have to blame others. You get rejected your publication and you read and you say, oh, those people are so stupid. Why they? And then you say, oh, they didn't understand what I was writing. They did not understand. I should write better because they did not. So you have to end up what we learn in high school, what I should do, not others. Because the stress is coming. You want your father, your neighbor, your girlfriend, your friends to do something and they are not doing, then you get always frustrated and upset and stressed. You have to figure out what you have to do, that those should do what you want them to do.
What a lesson for young people, people like Ramesh. When you get a PhD, come back, Ramesh. Then we can talk with her more. Okay. Thank you for joining, Ramesh. Let me talk with her other stuff, so you can listen. Yeah. Thank you.
Great